We believe that the use of flash nanoprecipitation to assemble complex nanostructures will allow researchers who do not have extensive experience in chemistry, engineering, or nanomaterial science to fabricate advanced drug delivery systems independently for their own specific applications and needs. The main advantage of this technique is the generation of large volumes of diverse nanostructures at high concentration, low polydispersity, and loaded with bioactive molecules, including small molecule drugs and proteins, all formed under sterile conditions.
Generally, individuals new to polymer synthesis may struggle because the polypropylene sulfide component is highly sensitive to oxidation by exposure to air.
Visual demonstration of this method is critical, as the multiple impingement steps are difficult to learn, because they need to be performed very quickly in succession.
To sterilize the confined impingement jets, or CIJ, mixer, work within a biological safety cabinet to submerge the mixer with all parts disassembled into 0.1 molar sodium hydroxide overnight. Reassemble the CIJ mixer and flow endotoxin-free water through the mixer using Luer-Lock syringes. Test the pH of the water and continue to flow water through until the pH registers as neutral. To prepare impingement solution 1, first weigh 20 milligrams of the synthesized polymer into a 1.5 milliliter tube. Then, add hydrophobic dyes, drugs, or other cargo. Finally, add 500 microliters of 100% tetrahydrofuran